So dear students, in this video I will make you able to understand a concept of Spirogyra sexual reproduction with reference to Scalariform conjugations and its details. So let's watch the video and get understand. You know, a sexual reproduction in Spirogyra, it is accomplished by a mechanism known as conjugation, or you can call it as a fusion, which is a very primitive method of isogamous type of a sexual reproduction in Spirogyra. It involves the fusion of entire content of two similar but unspecialized vegetative cells. When these cells get mature and get yoked, the conjugation process starts. According to phycologist Tiffany, there are certain changes takes place in pectose, which is a part of a cell wall in Spirogyra. So pectose get converted into a water soluble pectin compound, and such a water soluble pectin, it seizes in the conjugation thread. So seizing of conjugation thread, it takes place due to formation of pectin. In the view of Pesoni, lower concentration of nitrogen and high intensity of a light, it enhances the conjugation process. Sexual conjugation, it takes place toward the end of growing season. You have to remember this part. There are two ways by which conjugation occurs: Scalariform conjugation and lateral form of a conjugation. So when we talk about a Scalariform of a conjugation, or it is also called as a lateral conjugation, it is common in occurrence, and also it takes place in between the opposite cells of the two neighboring filaments. It is also known as a dioecious conjugation. It is seen in all dioecious species, that means in which filaments are unisexual. There are very rare cases in like as a monoecious species where dioecious conjugation may be also get recorded, but cases are very rare. Whereas the lateral conjugation, it is rarely found and take place between two adjacent cells of the same filament. It is also known as monoecious conjugation, that means in which filaments are bisexual. So when we talk about a very first type of a conjugation, that is Scalariform, or sometimes it is called as a lateral conjugation. Just have a look what are the events taking place during this Scalariform of a conjugation first, prior to go in much more details of all these processes. Have a look in this phenomenon. In Scalariform conjugation, two filaments of Spirogyra species, they come close together and lie side by side. The structure, it looks like as a ladder, so it is named as a Scalariform conjugation or H-shaped conjugation also. The tube-like structures that develop from each cell of two filaments lying together. The conjugation canal is get produced between two cells after fusion of the developing tube. It takes place, and the male gamete, it starts to travel, migrate toward the female, and of course the filament of one thallus or filament of one member or one species or one partner, it becomes empty, and other is get received with a zygote formation. So this is very interesting mechanism. The zygotes which are produced over here, they later on get released once the thallus structure starts to get decay or decompose. So parental filament undergoes a decay process, and of course zygote which is produced it after resting phase or when conditions become favorable, it germinates and gives rise to a new thallus. So you have understood this particular concept. I hope so. Once again, we'll try to look all these stages or consequences of a scalariform conjugation in shorter form. So single filament, if it is there, it has to be get a partner. Once they make alignment, the conjugation tube starts to get produced, as you can see on the screen. And of course, after that, the gametangia, which are possessing the gametes over there, they starts to show the fusion, but for that purpose there should be a migration of a protoplast of one of the parent, and generally it is a male one, toward the female. It is followed by the process of a fusion. Generally it is referred as a zygote formation. So zygote, once it is get produced, it gets entered into a mechanism of germination process. So how this scalariform of conjugation takes place, these details we are discussing now. It takes place at a night between the recently divided cells, and according to the Chapman, he says that this process of a scalariform of conjugation, it divide this entire mechanism into three distinct phases, such as the maturation phase, gametic union phase, and zygospore contraction phase. So now we will discuss about the details. Scalariform of a conjugation's very first stage or phase you can call it as a maturation phase. During this phase, the participating filaments of the same species of a Spirogyra, they come together. Okay. Morphologically, these filaments are alike. There are no visible differences present between them. Functionally, however, they are totally different. Physiologically, they are different. So, these differences become apparent only at the time of a fusion of a gamete. Otherwise, we cannot distinguish which one is male and which one is a female one. And during the night, this filament of two mating type, they come together, line up side by side and cell by cell, and the single filament, whatever maybe you can observe here, it does not move at all. The presence of other filament, perhaps it acts as a stimulus over there. So this is very important concept. So which one is a stimulating factor for staying together this filament? So the adjacent movement between the conjugating filaments, it may either brought by the localized secretion of certain chemical substance. Just now we have seen about the secretion of a compound, that is pectin, that is mucilaginous substance, or sometimes by inner surface tension differences. The close contact in between these two filaments, due to this steadiness or secretions, it induces in fact the firm attachment or firm is that a alignment of these two filaments together. So the sticking together here and there of a pair of a filaments pair, it is more often inducing the next phenomenon, that is formation of a papillae. As you can see in a diagram B, there are small protrusions are coming out. Generally they are nothing but the small papillary structures. So now we have to little bit think about how these papilla are get formed and what are the reasons behind that one in more detail. Basically they are formed on the recently divided cell. Just now I had told you that, which are naturally very shorter. There are differences of opinion with regards to development of a papilla. In fact, according to some phycologists, one of them is a Sonder, a short, very small bird-like papilla, it arises from one of the each pair of a opposite cell toward the other. As you can see, these are the papilla-like structures are there. Usually the papilla appear one of the conjugating filament. On touching these papillae, okay, once on touching this papillae, they stimulate or papillae stimulates the development of a similar outgrowth from the opposite and corresponding points on the other filaments. The papillae are thus in contact from the very beginning, and there is some other view also that they suggest that the papillae simultaneously grow. This papillae, they simultaneously grow from the young and recently formed cells of both the conjugating filaments. as in case of a diagram B, you can see from both the filaments these conjugating papillae they are starting to get developed. So there are differences of opinions are existing, and in fact it has been get proved also. Now when we look about these papillae, in consequent phase papillae they starts to get elongate to form a conjugation processes, and the conjugation processes of the opposite cell they meet very immediately to form a conjugation tube. So these papillae further get converted into the conjugation tubes. The conjugation tubes, of course, the latter it starts to get elongate and push apart the two conjugating filaments. So this happens due to the formation of a conjugation tube or conjugating tube, pushing away the filaments. And of course, due to this what happens, a ladder shaped type of arrangement of filament it starts to appear. According to the Lloyd stated that the conjugating or conjugation papillae of the female cell is usually shorter and thicker than the male one. In some species of Spirogyra, the male papillae they form the entire conjugation canal. So it's really very interesting mechanism that how conjugation papillae gives rise to conjugation canal. The development of this papillae according to the result of, you can say that a contact stimulus theory, or you simply call it as a thigmotropism, and thus it is compared to the rhizoid like developments. There is one theory, this is the stimulus theory. Others hold that they are the result of a chemical stimulus. So the development of papillae is either by means of a thigmotropism or it may be by means of a chemotropism also, by means of a chemical stimulus in fact. The Grot reported that there is a species of a Spirogyra known as Spirogyra majusculae, and it shows the growth of a conjugating papillae, or you can call it as a copulation tube also, is controlled by it is controlled not only by the thigmotropism stimulus but probably by a interaction between hormone like chemical substances which are migrating by diffusion from one filament to the other, depends upon the single division of the cell. So it's really interesting, and therefore the freshly agglutinated compound or agglutinating filament, freshly agglutinating filament that do not yet consist of a gametangia. It doesn't consist of gametangia, though they are establishing the papillae, but none of the cell we can say that it is a male gametangium consisting and female gametangium consisting. So these cells must be termed as, now a special terminology, these cells must be get termed as a gametangia, or not gametangia, excuse me, it should be a progametangium. The structure which will be further get developed into the gametangium is referred as a progametangium. All progametangial cells of the conjugating filament that undergoes a single cell division to form gametangia, and the gametangial cells united by conjugation tubes store of course a rich amount of or abundant amount of the starch as a food material over there, and the nuclei and nucleoli what may be are present in that particular protoplasm, they diminish in size, and there is of course also observed that the decrease in the permeability of a such gametangial cells it takes place over there, and of course the osmotic pressure of the cell sap is also dramatically get decreased. So these are the some important use that a processes taking place during this particular stage of a maturation of Scalariform conjugation. Now with the completion of a conjugation tube, the double wall which is present between these tubes, okay, that starts to get dissolved by certain enzyme actions, and a continuous passage it starts to get established over there, and it is referred generally as a conjugation canal. So such a conjugation canal which is developed over here, just now we have seen the certain reasons behind it, why it, or why and how it may be get established, that is by means of an enzyme action. So it is formed in between, of course, conjugation canal, it is formed in between two opposite cells. It is connecting bridge actually. The arms of the two of protoplast, especially protoplast means the protoplast of a gametangial cells, which extends into a canal, come in a direct contact of the other. Thus, the two protoplasts, they remain in contact from this is that a moment onward. They are always in contact through this particular common passage. With this particular phenomenon, a phase of a maturation, or is simply called as a maturation phase, is get completed. So dear students, we'll move ahead little bit now to understand what happens after the maturation phase with the Spirogyra during Scalariform of the conjugation. And the mechanism or that phase is called as a gametic union phase. During the gametic union phase, the protoplast of the conjugating cell, now which is generally called as a gametangia, it forms gametes. During this phase gametangia, they are get developed or they are produced. The gametes are formed singly. The protoplast which is observed over there, in this case of a gametangia, especially the protoplast which is destined to function as a male gamete, it starts to get shrink from parent cell wall without losing a contact with the other female protoplast. As you can see here, the protoplast, it start to get shrink out. One protoplast is starting to shrink out, and generally it is nothing but a male gamete actually. The contraction of this particular protoplast, it is brought about by removal of a water later on by means of a contractile vacuole. So role of a contractile vacuole is very, very important in this particular stage. The rounded male protoplast, the rounded, later on it becomes little bit rounded, that protoplast, it starts to migrate through its a respective conjugation canal slowly, and it joins, later on get fused to its partner protoplast of a opposite cell. Now that partner protoplast of a opposite cell we can consider here as a female protoplast or female gamete. This movement of a male gamete is often referred as a amoeboid movement. Okay, male gamete toward the female gamete movement is called as a amoeboid movement. But its mechanism of a amoeboid movement, it is little bit obscure and it is not yet get explored in more detail. So possibly the contact between two protoplasts having been established by the dissolution of a intervening wall between the conjugation tube, and their fusion and rounding may be get cheaply due to the surface tension. That may be one more theory over there, and this happens as you can see in the diagram E. Rounding takes place, it happens due to the surface tension, and a lot of factors may be there. It is only after the arrival of the male protoplast that the female contacts from its cell wall, and the two protoplasts basically fuse over there, which involves the fusion of a nucleus to nucleus and fusion of a cytoplasm to cytoplasm. So such a way, the fusion of two nuclei in certain cases has been recorded that it is little bit delayed for a while, and after that delayed time duration, that may be get fused. So that may be in certain species have been get reported. So when we look this particular mechanism, it ends with the formation of actual zygospore. So my dear friends, I hope that you understood what is the importance of a gametic union phase. It is not straight away that the two filaments comes together and they establishes a contact bridge and simply straight way that the male gamete is getting migrated or moved toward the female and fusion takes place. It's not like that one. There are many events going on and taking place. So as you can see here, it is a zygospore now. Let's have a look about how it looks under the microscope. Of course, this is the stained slide over there. So gametic union phase, it takes place in such a way that one of the cell is considered as a male gametangium, from which that protoplast by means of a amoeboid movement it migrates over there, and that migrating protoplast which is received by another cell is generally referred as a asexual or female gametangium, and this movement of a protoplast you can see here in taking place from by means of a so-called amoeboid is that a mechanism over there. Have a look for now the next step of a sexual reproduction in especially scalariform of a conjugation. So generally it is referred as a zygospore contraction phase. So this is the third and last stage of a scalariform conjugation, basically zygospore contraction phase. You know that the united protoplast or protoplasmic masses of the two filaments or two gametes, now they are producing a zygote like structure. Generally it is now referred as a zygospore. So the last phase to be get described is that the zygotic contraction. Just now we have seen that when zygotic contraction it takes place, which is brought about by a further action of the contractile vacuoles basically, and during this phase the contractile vacuoles of the zygospore, basically the protoplast of a zygospore's protoplast, it starts to lose more water. So this zygospore, which is getting now contracted, that is simply due to the loss of a excessive water over there. Consequently it start to get shrink away from more from that particular cell wall, and it looks like as a little bit rounded and oval structure. In fact, after that, a new thick multi-stratus brown colored wall is started to get developed there. Basically it is multi-stratus. That means there are three stratas are actually there, or three layers are there. Usually it is differentiated into the three layers, as you can see on the screen. The name or terminology which are labeled here, that is a little bit old terminology, such as the extremely is that a outermost, it is referred as a exospore. Basically it is a exosporium. Middle one, it is a mesosporium, mesospore, here it is labeled, and extremely innermost one, it is called as a endospore or endosporium. Now if you look here, the exosporium or exospore you can call, it is the outermost very thin colorless layer of the zygospore wall. Please remember the color, overall coloration, it is due to the all three strata to the zygospore. But when we talk about the individual layer of this wall, outermost one is called as exosporium, and it is a colorless layer of a zygospore wall. Of course, chemically it is composed with a cellulose, and when we look about this particular chemical composition of a exosporium, I'm sorry, cellulose is there along with a pectin compound also. So more often pectin is considered as a predominating compound over there, and of course it is a smooth, but sometime in certain species this exosporium has been recorded as a sculptured, but in most of the cases it is a smooth walled. Now let's move toward the middle layer, that is called as a mesosporium. It is considered as the thickest layer amongst the all the strata. So it is the thickest middle layer of the zygospore wall. You know, there are certain reactions like as acetylysis, and in fact certain enzymes, they may be get act upon. So such a acetylysis and enzyme reactions are get opposed or resisted by this zygospore. This is simply happen, it is presumably said that it is simply due to the sporopollenin, which is a chemical compound present in this wall or in this layer. This layer, it appears pale yellow to chestnut brown. Usually it is with dehiscence line. It possesses little bit dehiscence lines. You can see here the line dehiscence. This is the actual, we are talking about this particular layer. Okay, more often it consists of dehiscence line, and it may be more smooth, but in certain cases it is having sculptured outer surface also. So that depends upon the species to species, such minor changes, minor differences may be happen. Now if you look toward the extremely innermost portion, that is called as a endosporium or endospore. It is referred as a thin colorless zygospore wall. Chemically it is comprised with a cellulose in nature. And now friends, this overall structure of a zygospore, if you want to describe in shorter form, it is the green zygospore, which is usually ellipsoidal in shape or ellipsoidal in form. Rarely it occurs in a ovoid or lenticular in appearance. the structure later on toward the maturity. It becomes a little bit darker. Earlier it was a green, later on it becomes darker. As the individual chloroplast it starts to get disappear, this darkness appears. Okay, the male chloroplast generally uh, it starts to get disappear first from this entire zygospore. You know that protoplasmic fusion has been occurred in previous stages, and male and female cytoplasmic protoplasmic content is there. Now among that the first one which is get disappeared, that is a male chloroplast over there. That's why the coloration it changes from green to the darker one. And later on this zygospore is now capable for resisting the unfavorable conditions. It becomes like as a resting spore or resist spore, and it appears to be brownish or red in color, and of course it becomes a little bit densely filled with a starch and oily content over there. So this is what about actual zygospore which is developed as a result of its contraction phase over there. So my dear friends, I hope that you have understood this third but very very important stage in this Scalariform conjugation of a Spirogyra. This is the very beautiful slide photograph over there, in which you will notice that zygospores are produced in one filament, and second filament is having blank, you can say that spaces over there. So the cell wall of a original filament which is possessing that zygospore, it later on get a decay due to the adverse conditions, and of course through that zygospores are get released. And once they get released, they are of course a resting spore, and therefore after a period of a rest, or you can say that after getting a favorable condition, once they get a favorable condition, they returns into the metabolically more active phase, and through that as a result of a germination gives rise to a new filament, and in this way the Spirogyra it will be established as a new life actually. My dear friends, toward the use that a concluding remark of this entire session of a discussion, you have to remember certain points, facts analysis you can call, regarding the gametes and zygospore basically. So what we can say, what we can conclude about this particular aspect here, to please carefully watch this particular segment of a video. So gametes, the entire protoplast of a Spirogyra act as a gamete. They are known as aplanogametes. Aplanogametes, they are formed in gametangia, which are formed at the end of growing season of a Spirogyra. Whereas zygospores, the zygote in Spirogyra is generally known as a zygospore, and they are diploid in nature, and they are the result of the fusion of a male and female gametes. The zygospores are the only diploid stage in the life of a Spirogyra. This is the only important line among the entire discussion actually. There is a diploid stage in the life cycle of a Spirogyra. If it is asked, it is nothing but a zygospore. It has a thick cell wall, just now we have seen. It possesses three layers, like as the exosporium, mesosporium and endosporium. It remains dormant for a longer time till conditions not become favorable, and later on it get germinate. The mechanism of a germination, how that zygospore get germinated, it is not that much straight way it germinates. It needs much more use that changes, metabolic shifts are there, and out of that the zygospore it undergoes a meiotic division, then reductions happens, 4 haploid nuclei get developed, followed by that 3 are get degenerated and only one it gets survives. And what happens with that survived nuclear content, that part is a matter of another discussion. So I'll make a separate discussion on this particular part in a series of Spirogyra reproduction, and it will be the one very beautiful use that a video of a understanding, and I'm making sure that this particular video it will be cover the entire aspect of a maturation of a zygospore, followed by the germination stage over there. So you will get link for that particular video in a description box also. So my dear friends, these are some important facts related with the gamete and zoospore formation, and of course you have to look toward some another important aspect also regarding this Spirogyra, especially Scalariform conjugation, and how it is related with this sexual differentiations in fact. So when we talk about the gamete, just now we have seen that the gametes in Spirogyra, they do not show any kind of a morphological differentiation, and therefore they look similar in size and form, and hence the term what is you can see on the screen is called as a gametes or isogamete type. So they are isogametes which differ only in their behavioral pattern only. One of them is active, we generally referred as a male, and it moves from one filament to the another, where the second gamete, that is female, is awaiting for it. The slight distinction of sex between the gamete is accompanied more often by a sexual differentiation between the filaments. And the protoplast of all the cells of one conjugating filament, it act as a male gamete, and those of the other as a female gamete. These filaments are generally called as a unisexual filaments, or is simply called as a species is a dioecious. Really, this is one of the interesting fact about the Spirogyra sex differentiations. The filaments that gives up the own protoplast generally is referred as a male, as you can we have already discussed it, and one which receives it is generally called as a female. So despite this slight distinction of the sex during the sexual reproduction in the Spirogyra, it is generally referred as the sex of the Spirogyra is generally referred as or generally considered as a isogamous type of, you can say that, a sex over there. So because there are no other visible differentiations have been recorded in terms of a gametes, and in fact in terms of a conjugation filament also. So only these are the certain distinguishing points. Accordingly we can say it is a male, and with the second one with the female. So generally what I can conclude, the isogamous is the condition of a sex of a you say that is Spirogyra during Scalariform conjugation. So this is the concluding remark you can call about the gamete and sex of the Spirogyra during Scalariform conjugation. So my dear friends, thank you very much for watching this video. If you are having any suggestion, you can keep it into the comment box over there, and thank you very much. Keep watching for the further video in which you will get an information about the next process of a conjugation called as a lateral conjugation. So thank you very much.